Good morning. Thanks everyone for joining this presentation. My name is Lanie Galland from the University of Nevada, Reno, and together with Sarah Bisbing and Tom Parchman, I'm investigating the role of history and environment in shaping fragmented distributions of the emblematic fire pines of the coastal Pacific slope. So as you can see, the title of my presentation is just a bit different than our original title. So today, rather than presenting a final data set, I will be describing preliminary results as a launch point upon which our other analyses are building. So pines are among these most ecologically and economically important plants on Earth, and they occur in these really large ecologically variable ranges, playing an essential role in structuring forest ecosystems. They typically occur also in large genetically contiguous populations that exhibit little genetic structure connected by high levels of gene flow from wind dispersal. Quite a bit of diversification in Pinus has been recent, as seen towards the top of this phylogeny in the O. carpe. Here, however, we focus in on the trifolae section, which includes the North American hard pines, and then we're going to focus even more on the attenuate subsection, sometimes grouped together with subsection australis. Now, the clade with these three species, which include Pinus muricata, Pinus radiata, and Pinus attenuata, is a bit different. So from previous data, we interpret this divergence time from the ancestor in this clade to the middle Miocene 14 to 15 million years ago. So in contrast to other widely distributed pines, these species are found in highly restricted ranges and severely isolated populations, and they form this basal monophyletic group. So it's hypothesized that these three species experienced massive range constriction during the changing geological epochs, eliminating the population continuity in response to both physical and climatic barriers. They presently occur only in a few severely fragmented populations, with P. radiata and muricata limited to foggy maritime habitats along the coast, and attenuata scattered in stands extending from the coast inland into some subalpine habitat. Little to nothing is known regarding the levels of genetic differentiation among species and across populations, or how genetic variation is structured across the environmental and geographic space. So climate models predict extraordinarily high mortality for the evergreen trees of Western North America, and these range-restricted pines may be particularly at risk. Indeed, these species represent some of the most range-restricted pines on Earth, with 2 listed as threatened and endangered by the IUCN. So conservation and management of these populations will require a baseline understanding of how genetic variation and differentiation are structured across the landscape. Also, the historical processes that may have shaped present-day structure, if we see some, and the potential for local adaptation to environmental variation. So here I sampled throughout the distribution of all 3 of these species, including island populations, and we generated dd-RADseq data at several thousand single nucleotide polymorphisms for each species. Now, our goals with this project included estimating genetic diversity for comparison with other pines, including those widely distributed species. Now, are diversity levels lower in these range-restricted isolated species? And next, we plan to quantify genetic structure across spatial scales to characterize how genetic variation is distributed across the landscape. So given the disjunct distributions, do we expect population structure within species or uniformity given wind pollination and patterns in other conifer species? And finally, we will analyze the influence of fire, soil, and climate on patterns of genetic structure, and we'll search for genetic signatures of local adaptation. So today I'll be discussing recent preliminary results as relevant to our first objectives. And for the purposes of this talk, I will discuss preliminary results for the 2 most range-restricted pines in this study, muricata and radiata, as seen in panels B and C, respectively. So first, we see our muricata distribution and sampling maps on the left and the center, respectively. And right away, we see substantial spatial genetic structure in this species. The lower right cloud of individuals all originate from the northern coast of California, with the red cloud just above in PC space representing the central coast. Now we have next we have the more southern mainland populations in green and brown, and finally we have the island populations on the left side in PC space. So immediately we can identify 2 focal groups, the north coast and the island populations. So in the next 2 slides, I'm going to be zooming in on these individuals. So here I've analyzed the North Coast individuals independently, and we see some fine scale structuring across the landscape corresponding to a latitudinal pattern. In the south, we have this PR site constituting its own purple cloud and PC space. Moving north into a more continuous range of trees, we have the blue and brown SP and FR sites, followed by the green and orange PA and NR before finally going all the way north to this red Patrick's Point up near Humboldt. Now, oddly, we do have this pink population corresponding to Sea Ranch, kind of in the center of the North Coast, where some individuals group closely with green and orange, as would be expected, and others group with the red of Patrick's Point. And we're working through a couple possibilities as to this pattern. But overall, we see some fine scale structuring despite a fairly continuous range along the North Coast. Now, if we zoom in on the populations of Santa Cruz Island off the coast of Santa Barbara, things are even more interesting. So first, let's take a look at the map scale here. You can see sampling sites really are only 5 to 6 kilometers apart. When we look at the PCA, we have this remarkable structure on the island itself. So how is this relating to the demographic history of mainland populations? Are we seeing a metapopulation type dynamic or multiple colonizing events? These are all really fascinating questions fueling our ongoing analyses. So now we'll shift to the Monterey pine, Pinus radiata. Now, if you recall from previous maps, there are only a few naturally occurring stands of radiata. And once again, we still have substantial structure. Mainland populations group together nicely along PC1, with island populations grouping to the right. So from here, we decided to zoom in on Isla Cedros, shown here in green and orange. Now again, let's take a look at the map. We have a very small scale here with populations only about 10 kilometers apart. And again, we have very fine spatial genetic structure from these two groups of trees. Now for all of these preliminary analyses, we see pronounced spatial genetic structure range wide, and we see really striking patterns on the islands. So how is this occurring? Does it have something to do with fire, other environmental variables? So next, we investigated genetic diversity. Now, is diversity reduced in range-restricted pines occurring in small, isolated populations? Well, in this table, we can compare a few other Pinaceae examples that used comparable methods for nucleotide diversity. We might expect that these wind-pollinated species will maintain relatively high diversity as is found in some of these other conifers, or we may expect lower diversity because of isolation and fragmentation. But we don't. In fact, we see diversity higher than in all of the other conifers listed here, with the exception of white spruce. Is this surprising? Well, we really aren't sure. It's certainly surprising given the isolation and fragmentation characteristic of these species, but it's not quite as surprising given allozyme and mitochondrial DNA findings from the 1980s. So finally, I analyzed Tajima's D, where positive values indicate population contraction, like a bottleneck, etc., and negative values represent population expansion. So here, all values were negative across all of our sampling sites in both species. Now, the consistently negative estimates suggest population expansion following a bottleneck, inconsistent with the idea that these populations are contracting. Now, this supports previous research on radiata by Connie Millar in the late 90s, that these species have persisted in a metapopulation type dynamic over the late Quaternary. Population differences are thus likely to reflect frequent founder and bottleneck events. Now, for these preliminary analyses, we see pronounced and just remarkable structure within Muricata and Radiata, particularly on islands. Further, we did not find evidence of a reduction in nucleotide diversity and instead found abundant standing variation, suggesting that these two populations are not contracting after all. Now, this data only on these two species has opened the door to a number of additional analyses, and I can personally tell you that attenuata is no different. Now, moving forward, I'm interested in how climate, soil, and fire have shaped spatial genetic structure, and what environmental variables are shaping local adaptation, differences in soil water capacity, other environmental variables, and importantly, how maladapted might these species be under climate change projections? Can we model species mortality, expected shifts in distribution? We're very excited to move forward with this data. And with that, I'd be happy to answer any of your questions. Thank you for your time and consideration today.
Hello and welcome to my talk. I'm Emily Schumacher and this is Butternut Throughout Space and Time, Examining Range Shifts in a Rare Tree. So butternut, also known as juglans cinerea, is over to the right-hand side. You can see there's some lovely butternuts below. It is a tree species that is native to Eastern North America and is also in general rare. It is a non-dominant canopy member. It's also heavily threatened by a few factors, one of which is an invasive fungus that causes a pathology called butternut canker disease. I've highlighted a butternut trunk here with some cankers, and this leads to total tree mortality. And unfortunately, it's infected the entire range of butternut. Also, most of the species habitat has been converted for human land use, so it's also lost quite a bit of its range. It's also generally found in cold and wet areas, which eventually might be threatened by future climate change. So, in order to understand how the species could potentially respond to future changes to climate, we first need to understand how past range shifts have occurred in this species in response to climate change. So in general, we're thinking about protecting rare species. We need to make sure that they are genetically diverse because this allows species to respond to environmental change as well as protect their populations from pathogens. So this species was in general thought to have lost a lot of its genetic diversity due to things like butternut canker disease and habitat loss. However, a previous study in 2010 from Hoban et al. Identified that genetic diversity was reduced on a south to north cline in the species range. Also, there was identified to be significant structure between New Brunswick populations and all other populations throughout the species range. In general, these were thought to be due to glaciation being the biggest impact on the genetic diversity and structure of the species. However, Hoban et al. In 2010 did not fully sample the northern range of butternut. And in order to understand if its genetic diversity is truly reduced at its northern latitudes, we need to make sure it wasn't just due to the small population sizes that were included in the initial analysis. Also, if we want to understand the past movements of the species, we can't just look to genetic data. Genetic data generally is shaped by multiple evolutionary influences, so unfortunately it can't tell us everything we need to know about past range shifts. So we need to include more direct observations of things that were happening in the past. And we can do this by adding different types of data, one of which is fossil pollen records. Pollen can be preserved in rivers and lakes and sediments for a long time. And then when you pull them out, you can date them depending on what sediment layer they were in. And this can help to determine butternut or any species occurrence. So you can see down here, this is butternut records from about 1,000 years ago. Also, we can take what we know about modern species distribution. You can see I've modeled habitat suitability for butternut in the modern era, and then we can push this into past climate conditions, knowing what we know about the past and what climate would have been like during different periods. So in general, if we want to better understand how the species has moved around and what its genetic diversity and structure looks like in the modern day, we have to add northern individuals to our genetic analyses. And then we also have to include fossil pollen records and hindcasting species distribution models, which will all help us understand how butternut has moved in the past and also the current influences of modern climate. And this will help us to better protect and preserve the species into the future. So first of all, our genetic methods included adding 757 individuals to the initial 1,004 individuals from Hoban et al. 2010. And these individuals were mostly sampled from the northern range of the species range. And then we were able to measure genetic things like genetic diversity and structure. So looking at genetic diversity patterns, if we expected glaciation to be the biggest determining factor in this species' genetic diversity distribution, we'd expect it to be reduced going from south to north. And this is because, or it's hypothesized to be, because as individuals migrate into newly available habitat following glacial retreat, fewer and fewer individuals will be able to colonize those environments. And as population sizes get smaller, you lose genetic diversity. And this was observed and established in 22 hardwood species in Europe. However, it hasn't been as easy to establish the same pattern in Eastern North American tree species. If we expected modern climate to be a bigger determinant of butternut's genetic diversity distribution, we'd expect it to show something along the lines of the central marginal hypothesis, where as you get closer to the center of the range of a species, you expect it to show more genetic diversity because that's the most optimal habitat. Whereas in cases where you get farther away from the center of a species range, we expect it to be less and less suitable habitat, which leads to fewer and fewer individuals being able to tolerate those conditions, which then leads to reduced genetic diversity. So looking at our new results with the newly included individuals, we actually see that the central marginal hypothesis seems to be better representative of what's going on with butternut. So instead of seeing a linear decrease in genetic diversity going from south to north in this population's range, we actually see more of a quadratic regression being best fit to these data, with the most genetic diversity towards the middle latitude of the species range. And this is complemented by a really nice linear regression between genetic diversity and the distance from the range edge. So we see that the farther away from the edge of the range, the more genetic diversity you have. Now, as a note, these regressions are only significant without the Wisconsin populations, which suggests that those are even affected by different and new modern conditions for this species. Now, if we expected genetic structure to be most shaped by glaciation, we would see that individuals that shared different environments or shared the same environment during the last glacial period because they were pushed there by glaciers, we would expect them to show the most genetic similarity. And this was exhibited in Hoban et al. 2010, which found that New Brunswick populations were really differentiated from United States, Ontario, and Quebec populations. So even though New Brunswick populations may be closer to Quebec or Ontario populations, those Quebec and Ontario populations showed the most genetic similarity with United States populations that are more currently distant from them. And so this suggests that the New Brunswick populations were separated into a separate glacial refugia during the last glacial period from all of the other individuals. And so we see this supported with the newly added individuals as well. We can see here New Brunswick clusters very distinctly from Ontario, Quebec, and United States populations, which is supported by a principal coordinate analysis, which shows New Brunswick individuals cluster very closely, while all other populations cluster away from them. And there's a little bit of difference differentiated by PC2, but it's not really regionally specific. So in general, now if we're looking at the past evidence, we want to look at pollen methods, or pollen data points. So we downloaded pollen records from Neotoma and then mapped them in 1,000-year increments, 21,000 years ago to present. And then we used world climb variables to model butternut's distribution in ARI using boosted regression trees. And then using the output habitat suitability maps, we projected it into past climate conditions using paleoclimate variables. Now, looking at our maps, we can see that 21,000 years ago, so around the last glacial maximum, we see a really distinct area of suitable habitat, very bright green, in the southeastern US. Now, there's also a strip of suitable habitat next to a glacier off the coast of Nova Scotia. Now, that's an area that looks like, hey, it should be underwater. Well, during the last glacial maximum, the sea levels were much lower as everything was kept in ice. So there was an area of suitable habitat where the shoreline would have been much farther out into what is currently the ocean that butternut could have persisted next to that glacier. Now, there's no fossil pollen evidence, and that's likely because we simply can't get to sediment that's in the ocean, and also it would have been likely pulled away. So we don't have fossil pollen evidence of a population persisting in that area, but it's possible it could have. Now, going further or closer to the present, you can see that there's still a distinct differentiation between southeastern US and up near Nova Scotia, and we see that the pollen records start to support that differentiation between up near New Brunswick and then everywhere else in the United States. So just some caveats about our data sources. Past data is not perfect, Pollen records are not always indicative of organisms or populations living there because they can be blown from anywhere. However, it does give you a general idea of where organisms could have been. Also, in general, species distribution models assume that we are modeling the entire niche of a species and that we're including every data point, which isn't probably the case. Also, in this analysis, we assume that the niche of butternut didn't change in the last 21,000 years. However, we did see multiple factors shaping butternut in general. Genetically, we saw modern climate impacting it significantly, but structure was really seen to be impacted by past glaciation. And this was supported by past species distribution models, as well as pollen records, with those two separate refugia near New Brunswick and the southeastern US. In general, when we're looking forward, we can see that butternut is indeed influenced by modern climate. And what we don't know is if the levels of genetic diversity it exhibits right now are enough to respond to climate change. Also, butternut does appear to have been able to shift its range in response to past glaciers, but is this fast enough for it to respond to future environmental climate change? So now I will take any questions. Thank you so much for coming to my talk.
Okay. Hi, and thanks for coming to my talk. My name is Katie Uckele, and I am currently a fifth-year graduate student at the University of Nevada, Reno. I'm excited to present here at the Forest Genetics Student and Postdoc Symposium and to introduce some of my newest research investigating the drivers of variation across the juniper hybrid zone in Western Nevada. When diverged lineages come back into secondary contact after a period of allopatry, oftentimes species boundaries are tested. If reproductive isolation is incomplete, hybridization often occurs. Not only does hybridization have consequences for gene flow among species, but it can also dramatically reshape phenotypic variation across the landscape. Reshuffling of certain key phenotypes, like plant secondary chemistry, can have cascading effects on interactions, biodiversity, and ecosystem processes. My work aims to disentangle the pathways connecting genetic, geographic, and environmental variation as they directly and indirectly shape the phytochemical landscape. The focus of much of my research is centered in Western Nevada, where three distinct juniper lineages are experiencing secondary contact. Juniper is an underrepresented genus compared to other Western conifers in terms of their population genetics, and these three junipers are no exception, despite occurring over large geographic areas in the West. In Northern California, Oregon, and extending into Washington state, Western juniper occurs at mid to high elevations in the Eastern Cascade foothills. Its sister species, Sierra juniper, can be found growing at even higher elevations throughout the Eastern Sierras. Finally, Utah juniper can be found in the broadest geographic range and at even broader altitudinal ranges across the Great Basin, including Colorado, Montana, and Arizona. In addition to being an important source of habitat and food for many desert wildlife, these Western junipers host a highly specialized caterpillar community whose natural history is poorly understood, but whose abundances and distribution may be affected by hybridization. For the study and to produce the following results, I sampled 25 populations representing 326 individuals and generated over 8,000 genomic loci utilizing a ddRAD-seq approach. First, I utilized this genomic dataset to resolve the evolutionary history of these species. Perhaps due to integration, previous attempts to reconstruct their history didn't align with the morphology of these trees. Utilizing thousands of loci, the phylogeny I recovered is consistent with morphology and also confirms that Sierra and Western juniper are distinct evolutionary lineages. A PCA of genotype probabilities confirmed hybridization among these taxa, as indicated by the large cloud of gray points intermediate between our parental clusters. Though I wanted to focus on the hybrid zone for this brief talk, I should mention that within each of these 3 juniper lineages, population genetic structure is extremely low, consistent with low genetic structure seen in many other wind-dispersed conifers with large geographic ranges. Structure analysis, assuming 3 source populations, provided enhanced resolution of this hybrid zone and showed that most hybrids possess ancestry from all 3 parental lineages. Across the landscape, ancestry proportions are highly variable but can be predicted by geography. This result inspired me to consider whether patterns of phytochemical variation were similarly variable and complex across the landscape, which could have extended consequences for plant-insect and plant-animal interactions across space. To visualize patterns of chemical similarity between species and hybrids, as well as the variances within groups, I utilized ordination methods based on 163 GCMS features representing a full spectrum of terpenoid diversity. I attempted to isolate 2 aspects of chemical similarity, qualitative and quantitative, where qualitative similarity refers to a large number of shared chemical compounds among groups, whereas quantitative similarity refers to many compounds with similar relative concentrations among groups. First, to assess qualitative similarity, I based my principal coordinates analysis on binary distances calculated from the simple presences and absences of features. This ordination demonstrated that hybrid individuals shared a greater proportion of compounds with Sierra juniper than with the other 2 parental groups. However, despite this large degree of overlap, an ANOVA based on distance matrices confirmed that the sets of compounds expressed in Sierra and hybrids are distinct. Second, to assess quantitative similarity, I conducted another principal coordinates analysis, but this time based on Euclidean distances calculated from the relative concentrations of the chemical features. For each of the 4 groups, I calculated Euclidean distances between group members and the group centroid and used ANOVA to test for homogeneity of within group variance. The mean distance to centroid for each group is provided in parentheses, and the distribution of distances was significantly different for each group, with Western juniper having the smallest variance and hybrids exhibiting the most variance in compound concentration. This pattern of high hybrid variance is consistent with patterns of hybrid admixture, which are complex across the landscape, yielding a continuum of hybrid ancestries. That hybrids are qualitatively and quantitatively distinct from their parentals may have implications for communities which interact with junipers via chemical cues. But it could also favor the persistence of hybrids if novel hybrid phytochemistry provides adaptive potential. Finally, to identify the major drivers of phytochemical similarity across the landscape, I utilized a Bayesian distance-based structural equation model incorporating geographic, genetic, and environmental distances as direct and indirect predictors of phytochemical distance. I can discuss this model further after my talk if anyone is interested, but to briefly summarize the results, I found that geographic distance strongly predicts genetic distance among populations and weakly predicts environmental distance across the landscape. Environmental distance, in turn, moderately predicts genetic distance, suggesting that isolation by environment has played a role in structuring genetic variation across the landscape. Finally, while environmental distance appears to have no discernible effect on chemical distance in this model, genetic distance is a strong predictor of chemical distance. A caveat of this result is that we incorporated all populations across the landscape, which represent 3 diverged lineages, and perhaps zooming in on a single lineage may indicate a greater role of environment. Thank you for your time and attention while I present my most recent work investigating the drivers of variation across a juniper hybrid zone. The results of my work show that hybrid ancestry is comprised of 3 distinct parental lineages, and a continuum of ancestries exists on the landscape. Hybrid chemistry, similar to hybrid ancestry, is also highly variable, but qualitatively resembles cedar juniper more than the other 2 parental lineages. Finally, across parentals and hybrids, secondary terpenoid chemistry is largely determined by genetics, though geography and environment may play an indirect role. Feel free to ask me any questions during the break, but please don't hesitate to reach out on any of my other platforms if you have any questions about my research. And again, thank you for your time.
Hello, I'm Whitney Jackson. I currently work as a consulting arborist with Save a Tree Consulting Group. I'm happy to be here today presenting population structure within the hard maple species complex, a study I conducted during my graduate program in plant biology at Rutgers University in New Brunswick, New Jersey, which I graduated from about a year ago. A manuscript describing the study was accepted for publication this past March by Canadian Journal of Forest Research. I will give a brief introduction of hard maple species used in this study, then I will describe materials and methods, present study results, and follow with a brief discussion, conclusions, and future foci. The hard maple species complex is comprised of a group of closely related, highly variable, and taxonomically ambiguous taxa. Figure 1 shows 6 hard maple native range maps, each associated with one of the 6 species represented in the study. They are Acer saccharum, sugar maple, bordered in green, Acer nigrum, black maple, bordered in blue, Acer leucoderm, chalkbark maple with the orange border, Acer barbatum, southern sugar maple with the yellow border, Acer granidentatum, big-tooth maple, which is bordered in red, and Acer schuchii, cloud forest maple, bordered in purple. Hard maple species express a high level of plasticity, enabling them to adapt into environments that vary in elevation, hydrology, soil type, average annual and monthly temperature, and seasonality. Sugar maple is by far the most recognized of the hard maples. Images on the slide here are examples of the cultural, ecological, and economic value of sugar maple worldwide. Since the 1950s, popularity of sugar maple has driven research focused on increasing sugar concentration of tree sap, its quality, and yield to provide maple sugar producers with improved nursery stock. This is great for sugar maple, but has contributed to a literature deficit where its allies are concerned. Sugar maple has been identified as a species at risk in the face of rapid climate change by the United States Forest Service due to drought and temperature sensitivities. Figure 2 is important because it shows the expected trajectory of sugar maple under 3 climate change mitigation scenarios in the United States. The maple beech birch forest type indicated on the map by the light brown color, as you can see, becomes increasingly green, which indicates presence of the oak hickory forest type. What that means for the maple sugar industry is depicted by the image on the right, which shows a reduction in the production of maple syrup beyond the year 2100 as habitats become undesirable and trees are no longer able to thrive and produce. Unless we do something to mitigate the negative impacts of rapidly changing climates faced by sugar maple, it's bye-bye maple sugar. To help fill the void that exists in hard maple research and increase our knowledge of the genetic relationships between hard maple species, we designed a study that would compare genotypic profiles of available hard maple species, estimate divergence levels between study groups, and then determine whether the divergence estimations reflected a geographic pattern across the study groups. Table 1 summarizes study species, their provenance, latitude and longitude of provenance locales, and the number of samples used for each study group in our analyses. We included 2 populations of A. saccharum, one from Michigan and one from New York, 2 populations of Acer leucoderm, one from South Carolina and one from Texas, 1 population of Acer nigrum from Minnesota, 1 population of Acer barbatum from Texas, 1 population of Acer grandidentatum from Texas, and 1 population of Acer schuchii from Mexico. Microsatellites used in this study were developed by Graniuk et al. In 2013, and the 17 used are listed in Table 2 at the lower right of the slide. Genotyping analysis was done using capillary electrophoresis equipment and DNA size standard. GeneMapper was used to analyze allele calls. AMOVA paired population comparisons, routine in genetics, was used to access divergence levels between study groups, and the principal coordinates routine in genetics was used to visualize divergence levels and pattern among and within study groups. Table 3 summarizes results of the AMOVA pairwise population comparisons. Population codes are listed along the perimeter of the table. Values within cells represent divergence among and within hard maple study groups. Red arrows indicate values for interspecific divergence between populations of Acer saccharum from Michigan and New York, coded in the table as MIAS and NYAS, and Acer leucoderm from South Carolina and Texas, coded as SCAL and TXAL. Our population of Acer nigrum is highlighted in gray to note that divergence values associated with the Acer saccharum populations are also low. The greatest divergence was estimated between Texas barbatum and Texas grandidentatum and is highlighted in yellow. Principal coordinate analysis based on pairwise data in Table 3 helped visualize pairwise comparison estimations and is shown in Figure 4. Differentiation levels between populations are determined by distance between points on the ordinate plane. Interspecific groups are represented by the same marker type, and each interspecific grouping is within a red ellipse. As expected, populations sharing lowest divergence values are seen in closest proximity to one another, as shown in the Acer saccharum and Acer leucoderm groupings. Those sharing the highest divergence values are farthest apart, as seen between Acer grandidentatum and Acer barbatum, highlighted in yellow. Acer nigrum is highlighted in gray, again to note that it is closer in proximity to Acer saccharum populations than any other group on the ordinate plane. So our discussion here is a brief summary of the previous slides. Sugar maple has value and is at risk in the face of rapid climate change. And despite a robust research base focused on sugar maple, studies addressing species limitations of its allies are severely lacking. This is unfortunate because sugar maple needs help to make it through the projected climatic transition. Adaptive traits observed in other hard maple species may be useful to develop strategies that would increase sugar maple's chances for a sustainable future. Our results suggest that Acer nigrum is the closest genetic relative to sugar maple. This supports reporting of observed hybridization zones where the two are sympatric. I would like to point out that the study groups do not represent the full extent of variation within the hard maple species complex, these species, or this section of the Acer genus. Conclusions of this study were that genotypic profiles of study populations place study groups in distinct and separable gene pools, and that the principal coordinates analysis showed a geographic pattern that correlated with provenance of study species. Molecular studies with increased representation of the variation that exists within individual hard maple species and physiological studies that track adaptive behavioral responses are needed in the immediate future to lay the necessary groundwork for bringing the hard maple species into the modern era of plant science. For that, I'd like to thank you for listening, and I believe we're going to have some questions. Thank you.